wet lab scientist. I do viral host interactions using orthopoxviruses. And so I'm going to do a really quick overview of one of my favorite projects that we work on that I've affectionately titled Die to Live Another Day, because it's a battle between a virus and the host to control cell death. And so themes you'll see in my talk here is that cell death is good for the host, it's bad for the virus. So if the cell dies, the virus dies, and the host survives. So that we'll see repeated over and over again. And if you're not familiar with programmed cell death, that can sometimes seem counterintuitive. So if there's no cell death, the animal dies or the human dies. So, okay, so we really care about how we respond to viruses as a host. So I'm a really bad virologist in that I don't care about the virus very much. I like to use it as a tool because I think it's great in terms of, measuring how we as a host species respond so that we can better shape vaccines and therapeutics from there. So how do we detect the viruses and how do we balance this protective versus damaging response and use this to our advantage? So I use the orthopoxvirus model. I use this because there's really well noted members of this that cause severe disease, including smallpox and more relevant recently, monkeypox. And then we also have vaccinia, which is the vaccine strain of the smallpox vaccine that is very, very good at providing protective immune responses. So we know this has been in our population for a very long time, probably as long as domesticated animals have been around, with evidence of Ramses V succumbing to a pox-like illness. And then we also know that it's really, really good where we can harness it to trigger immune responses with some of the first vaccination programs ever being instituted with Jenner and the smallpox vaccine using cowpox. So some really high level overview. It's generally considered a brick shaped, which is kind of odd for a virus. It's a very large virus and it's double-stranded DNA. And within that double-stranded DNA, it's unique in that it replicates entirely in the cytoplasm. And to do that, it encodes a huge amount of gene products with average around 200 gene products. But most of these gene products are actually innate immune evasion mechanisms. So these are not essential for viral replication, but they are essential for blocking the host response. And so we use this as a model to study this balance between protection and inflammation. And so we're looking particularly at RNA pathogen-associated molecular patterns. We know that it's really important for the host to be able to sense unique types of RNA in an infected cell. And so we're looking at this protein here where the virus encodes this E3 protein. Now, E3 kicks on, people think E3 ligase. It's not E3 ligase, it's just the gene structure and how they end up naming the genes in this virus. So it has a traditional A-form double-stranded RNA binding domain, but then it also has this other domain that is not, for a long time was unknown its function. It's super well conserved amongst all the orthopoxviruses, and we know that it's absolutely essential for pathogenesis. You damage this domain at all, you go from a very, very lethal virus in a mouse to a virus that can't kill at even 5 logs higher of a dose. So there's a 3 to 5 log attenuation, even in direct cranial inoculation, which is absolutely insane by deleting half of a protein in the innate immune evasion group. The virus goes from pretty lethal to basically harmless. So it turns out that this is a Z-form nucleic acid binding domain. And what Z-form nucleic acid is, we know Watson-Crick type of helixes where you have the right-handed helix. That's what we traditionally think of in DNA and double-stranded RNA. But if you actually force a left-handed helix, it creates a zigzag backbone in the phosphate backbone structure. This is structurally very, very unique from our A-form or B-form DNA. So this was a putative DNA binding domain, but why does the virus have it? Why is it so well conserved? Well, it turns out that this is really important between a competition between the virus and the host, in that when the virus doesn't sequester this pathogen associated molecular pattern, it triggers sensing through a host protein that also has a Z-form nucleic acid binding domain. And that ZBP1 protein can activate a cell death pathway called necroptosis, where this is actually a very inflammatory cell death, where membrane gets disrupted and the membrane basically explodes. So we have this direct competition for Z-form nucleic acids, right? And when we take away the virus's ability to inhibit, within 6 hours post-infection, these cells literally just burst open. Has anyone seen Shrek and seen Fiona like blowing up the bird? That's what I kind of think of happening here, right? They're just exploding, which creates a whole new level of inflammatory signaling that we can look at. So I'm going to skip this part. We know that this is necroptosis because we see hallmarks of the necrotic death and the phosphorylation of MLKL. We went ahead and verified that this is biologically relevant because there's a lot of debate whether Z-RNA is actually important. And we take this and we know that normally this mutant virus that can't sequester Z-form nucleic acid doesn't kill the mice. But if we go through and delete any part of the pathway that is sensing or triggering necroptosis, this normally pathogenic virus phenocopies wild type and is just as pathogenic. So this is critical for host response. And it's also critical for the virus to be able to block it for this balance between viral and host coevolution. So how is it blocking the ZBP1? And I've kind of already hinted, this is a Z-form nucleic acid binding domain. We went and made a bunch of these point mutations to specifically disrupting the amino acids that bind to Z-form nucleic acid. And any disruption of the Z-form nucleic acid is as efficient or as attenuated as the full deletion. So this is really saying it's Z-RNA. And we looked this, and the E48A mutation, which disrupts Z-RNA, gets the same phenotype where it's inducing necroptosis, whereas our control mutant doesn't behaves like wild type, and we never get necroptosis induction. So we confirmed that this is Z-form nucleic acid by using a monoclonal antibody, and showed that we do get actual induction of Z-form nucleic acid. And this Z-form nucleic acid is actually RNase because it's resistant to RNase treatment and, resistant to DNase treatment, sensitive to RNase. So we have this model where E3 comes in and binds to double-stranded RNA, and we also binds to Z-form RNA. Both of these are essential for pathogenesis. It prevents a direct competition between the host and the virus for binding of this unique structural type of RNA, versus when we don't have it, the functional protein ZBP1 is able to come in, bind to that, and activate antiviral death pathways. So interestingly, we know that we've said this is super well conserved, except one. So this is, we domain swapped, but that's not super important. So this is our model where we come in, the virus undergoes transcription, we create Z-form nucleic acid, E3 normally prevents that, letting the virus and cell both survive, which ends up killing the host. We said this is super, super well conserved in the orthopox viruses. I kind of lied in that monkeypox is the one exception to this orthopox virus rule. Monkeypox is actually the only one that naturally does not encode a Z-form nucleic acid binding domain in this E3 protein. We've searched the genome, there's no other suggested Z-RNA binding domains in the virus. So how is it actually getting around this super well conserved need for a Z-form nucleic acid binding domain? And it turns out our preliminary data is suggesting that it is picked up a novel mechanism to inhibit necroptosis. So it's still producing Z-form nucleic acid, where our Z-RNA staining is really robust at 8 hours post-infection. It's still causing phosphorylation, which is the terminal activation of the pathway of necroptosis, but it's not ending up losing its membrane integrity. So it's not undergoing that cell lysis event. And so the virus is still able to replicate and grow fine, even though it's producing Z-RNA. MLKL, which is the last step of necroptosis, is getting phosphorylated, but we don't lose the membrane integrity, so the virus still survives. So we're currently investigating where along this pathway is the virus actually inhibiting, and what specific mechanism is it doing. So the only other virus we know that has this kind of block at this level is human cytomegalovirus, but we don't have any suggested proteins or functions in that. So we're undergoing a lot of screens to try to figure this out to see if we can create specific targets to block that viral protein. So we should care about Z-RNA because we've proven now that it is not just an arbitrary event that is part of a biochemical mistake in a test tube. We see biological significance in animals during virus infection in cell culture, where this is a real event that happens and it has biological outcomes in health and disease. We just are breaking through on what this is doing, but more than likely, this is not just regulating cell death during viral infections. This could be related to interferonopathies, autoimmunity, because we know that autoimmune disease is frequently associated with anti-Z-DNA antibodies. So we can use this model to kind of poke and prod what happens when Z-RNA is exposed, to understand the consequence, inflammatory and immune consequences, and try to break through some barriers in our understanding from there. So I want to, make sure I thank my collaborators, because this has been a long-term project going on. And then I also want to thank my lab. We're a brand new lab. Been here just about a year. This is actually my very first undergrad presenting on monkeypox at the Undergrad Research Symposium. So with that, I can thank everyone and turn it over.